Welcome to the BioNow, a weekly webinar series where we discuss health, wellness, and longevity. I'm your host for today, Alina, and this week's webinar we will explore how science turns into solution. Our speaker for this week's webinar is Dr. Minal Borkar Tripathi, the Director of Genomics at BioAro. Her Ph.D. work in medical genetics focused on the human genetic variation in indigenous populations. In addition, she has extensive experience in molecular diagnostics, and her research has been recognized with awards and presentations at various international conferences. And today, she will help us discover how knowledge of your lifestyle, genetics, and environment can help develop personalized picture of the person's health. Before we proceed, I would request all the participants to adhere to the following rules. First, please keep your microphone muted at all times as the background noise can be disruptive. Use the chat box if you have any concerns or questions or face any technical issues. We want to inform that this webinar will be recorded and shared in various social media platforms for the learning purposes. I also want to mention that your feedback is valuable for us and help us improve. So feel free to share it at the end of the session. I will appreciate it very much. Without further ado, please join me welcoming Dr. Minal to learn something exciting today. Over to you, Dr. Minal.
Hello everyone, good evening. So today I'm going to talk about the topic that is translational medicine and multidimensional clinomics. What's a translational medicine? It's actually the idea of moving medical research from the experimental platform to the hospital bed. Was originally put forth in Science in 1992. Afterwards, in 1995, Lancet published the first article using the term transformation medicine. This is the field which translates the findings of basic research into new technologies and treatment approaches, allowing patients to genuinely benefit from scientific and technology advancements. Translational medicine actually encourages mutual change, drives basic research with clinical treatment, as well as advances clinical treatment with basic research. So, if you'll see how this is the link between the exploratory research and application of the medicinal products resulting from such a research. So whatever in the laboratory, the work going on in the cell lines, it is successfully completed on the mouse models or animal model, and then it is conducted in clinical trials on human. So this is how in translational medicine, the research from experimental platform to it reaches to the hospital bed and benefits the patient. Over the last 5 decades, the amount of data and information in the biomedical research has exploded. At the same time, the needs and capabilities for computing have exponentially increased. So, to understand translational medicine, also we want to understand the different omics, and omics is actually information system which is involved in translation medicine, and then we can understand multidimensional clinomics. So this open molecular information system is a rapidly growing and powerful technology class, allowing scientists to share and analyze large complex data sets. To study different omics, first let's discuss central dogma. So, central dogma of molecular biology is classified now in two categories. One is classical central dogma, and a second one is modern. So, in classical central dogma, it's actually it includes DNA. When a transcription takes place, then the RNA is generated, and because of translation, then protein synthesis takes place. But in second category, we talk about other factors which controls DNA and the transcription. For example, methylation of promoter region of genes inhibits the transcription of DNA. Hence, gene is known as downregulated, and result of it, no protein is produced. So this is known as epigenetics. Now, epigenetics regulates the DNA transcription, which impacts RNA generation and protein synthesis. Ultimately, this protein production also helps in the function of different organs. So it impacts the function of the organ. Therefore, this modern central dogma includes two new components. One is epigenetics, and second one is metabolomics, that is the study of metabolites. So let's discuss in brief all different omics that we know so far. So first one is genomics. So genomics is the study of the entire set of genes in the genome of a cell or organism. Genome is constant in every cell of an organism. There are high throughput techniques which are used in the genomics to map, to sequence, and to analyze genomes. For example, DNA sequencing can be directed, or it could be directed, targeted, customized, or whole genome sequencing or whole exome sequencing can be carried out. Similarly, there are genotyping assays which is used to identify the single nucleotide polymorphisms. Then next is transcriptomic. Transcriptomic is actually the study of transcriptome, profiling of gene expression, or understanding the splicing mechanisms that occurs between exons and introns. Through RNA sequencing or microarrays, we can determine the downregulation or upregulation of gene expression and link it with different pathway analysis. After transcriptomic, it is proteomics. So proteomics is the study of the entire, sorry, switch off. Just a second, please. I think there is some interruption. Yeah, so I think it's fixed now. So we were talking about we were proteomics actually. So proteomics is the study of the entire set of proteins produced by cell. The proteome is dynamic and varies compared to genome. The set of proteins produced in different tissues varies according to the gene expression. The different techniques to study proteomes could be the extraction and electrophoretic separation of proteins, digestion of proteins into small fragments to analyze. Then next omics is metabolomics, where we study different metabolites production or determine their level, which is known as metabolomics. The metabolomics could be carried out using nowadays it's the advanced technology mass spectrophotometer to check the level of specific proteins and also to study the different antibodies, enzymes, or hormone level. Now coming to the epigenomics, in epigenomics we study epigenetics, that is the different factors which can regulate DNA and transcription process by hypermethylation of DNA, histone modification, or by microRNAs. If promoter region of gene is hypermethylated, then it is inaccessible for transcription factors to bind, which inhibits the gene expression. Similarly, histone acetylation results into packed nucleosome, which inhibits the transcription process, and then there is no protein synthesis. MicroRNAs are small RNA fragments which binds to messenger RNA and again inhibits the protein synthesis. Hence, these factors, which are regulating the DNA and transcription process, are known as epigenetics, and study of all these epigenetics is known as epigenomics. Then next omics is variomics, when we study the variation in the genome. For example, when we study copy number variation in DNA or ribotyping, then it is known as variomics. The variations are determined at molecular or submolecular levels. Next is phenomics. Phenomics is studying genomic components and phenotype character. It's actually the evaluation of morphological characteristics, biological process, and interaction of an organism with physical factors or environment. It is to study the close association of an organism with habit and habitat. The next is inomics. So various kinds of metals or minerals, all of them play important role either in terms of protein activity or enzyme activity. So when there is element profiling or quantifying the levels of minerals using different techniques, then this kind of study is known as inomics. For example, presence of iron, its role in hemoglobin synthesis. Also, we know the optimum level of sodium, potassium, phosphates is important for normal organ function in the body. Next one is pathogonomics. We will split this terminology in three different terms, that is patho is pathogens, gut, and omics. Pathogens means identification of microorganisms which is found in gut, and omics is if we are using different uh, technique. It could be expression or a shotgun metagenomics to identify those microorganisms in the gut. So the latest example is gut microbiome. The knowledge of pathogens or opportunistic pathogens in the gut help in understanding the dysbiosis of gut, obesity, colorectal cancer, or even depression by studying gut-brain axis. So in BioAro, we test gut microbiome using advanced technology. Analyzing gut microbiome will also help us in reversing the gut microbiota for our benefits or treatment. For example, by adding good bacteria using prebiotics and probiotics. This is also known as synthetic gutomics. And metagenomic is actually study of different genome or genetic material from multiple organisms, which we are using in BioAro. And it's the advanced technology. So you can see that there are several omics, for example, genomic, transcriptomic, epigenomics, and proteomics and metabolomics. Other than this, there are pharmacogenomics, where it's the studying the impact of genes on individuals' response to drugs. There is toponomics, study of cell and tissue structure. Kinomics, study of protein kinases, and trialomics, like study of human interventional trials from clinical trials, so and so on. So this slide is actually talking about application of multi-omics in single-cell genomics. So here we can see the molecules are DNA, RNA, protein, metabolites, and the measurement was using different techniques in genomic, in epigenomic, and transcriptomic. So the level of protein or the level of metabolites, those are measured using different assays, and then the combination of these proteomes, metabolomes, or genome, epigenome, or genome or transcriptome, applying those multi-omics approach, it could help us in clinical setting to uh, uh, benefit the patient. So what about all these different pieces, which are actually pieces of from genome information, transcriptome, proteome, metabolome? All these are different pieces, and we have to put all together to interpret and get a picture out of it which is meaningful and useful. So uh, in this the genomic application, we'll see that how it has helped in public health. So for example, evaluation or monitoring the health, for the diagnosis, or for developing the policies. Whereas how our body is coping with the stress, how the response is against the environment when it is not favorable condition. This kind of thing is interplay between the episome and genome in health and disease. This kind of study is known as study linking between episome and genome, which is more over about the environment and lifestyle, which is impacting the health of the patient. Now, coming to the longitudinal personal omics profiling, where we can see all different kind of omics, and I would say multidimensional omics approach is used. And then on the basis of this, there is one example here. It is given for weight gain and loss, where the protein level metabolites were uh, determined in plasma. Then genomic, the single nucleotide polymorphism profiling was done, transcriptomic was carried out, and the protein with the mass spectrophotometry was studied. And then there is another information we are getting from microbial abundance in stool, and then cytokines and serum. So all these informations are integrated together to get the to find out in the weight gain or loss how it is applicable or how it can benefit us. So there are different major health discoveries occurred because of this multidimensional approach. So, for example, it helps in biomarker discovery in prognosis, response to treatment, that is pharmacogenomic markers, then molecular profile based patient clustering, that is stratification, the risk prediction and diagnosis. However, still, uh, there are pitfalls. Some of them are in progress, and some of them are like it's promising. For example, polygenic risk scores, these are the things which are in progress by using all these different omics. In cancer, still the somatic variation and germline variation, these are some of the pitfalls. And in a non-invasive testing or an ICU diagnostic, these genetic testing or genomics and epigenomics is playing very important role. So when now the next thing I want to talk about is physiome, which is used to measure, for example, different biomarkers. It could be a measurement of blood pressure, or it's like a smart watch, or this device Apple Watch. Many of people are wearing, millions of people nowadays, and it is used for more than thousands of measurements each day. It could be for heart rate, respiration, blood pressure, or oxygen level. So the genomic data mainly could be used for assessment of the predictive risk. Also, this data could be used to incorporate in clinical trials. In healthcare, and also it's most of the in oncology, we are using this genomic data to see how it could help in the treatment. So for example, in cancer, this we can see that there are many levels of interaction: genome, epigenome, and transcriptome. So this can be measured by the different omic technologies, and this is the example of the cancer diagnosis. So where a correct cancer diagnosis is essential for adequate and effective treatment, because every tumor is involved in interactions with non-cancer elements, such as gene environment interactions, microenvironmental interactions, and those with the immune system. Intercellular interaction within the tumor environment, and intracellular interactions such as transcriptional regulation, gene co-expression. So signaling and metabolic pathways, as well as protein interaction. So we can see in cancer genetic itself, there are different interactions between the tumor and non-cancer elements. So this is the example of that. Now coming to the theranostic. So from transnational medicine data to theranostic. So we know about the medicine, now we know about the precision medicine, and now we are moving towards the theranostic. So theranostic is a term which is derived from a combination of the words that is therapeutics and diagnostics. In this emerging field of medicine, drugs, and or techniques, these are uniquely combined to simultaneously or sequentially diagnose and treat medical conditions. So this is the example where this is the example of radio tracer actually. So in cancer, radio tracers are used for both diagnosis and treatment. So radio tracer are compounds made of radioactive chemicals that selectively bind to a specific target in the body. A radio tracer consists of two pieces. One half is the biological side that travels through the blood to bind to a unique designated target, in this case, a type of cancer cells. And the other half is a radioactive component. So during the diagnostic scan, the radioactive component exerts a low amount of radiation that allows us to take picture of cancer cells onto which the radio tracer has labeled. So the same compound is used for diagnostic as well as for the treatment. This is the example of theranostic. So in BioAro, actually, our approach is to work extensively and explore all these different omics and integrate the data to help patients in predicting risk of the disease, early diagnosis, monitoring, and timely treatment. So around year 2015 to 2016, many scientists and researchers realized that they have plenty of heterogeneous biological data from different techniques and assays. Idea to combine all these and put all together to discover new biology. So now, as we grew a lot in our understanding, and there are industries working on developing different platforms for data integration and evaluation. So we have a lot of information that is gathered through all these different omics. Then the important part is data integration. So data need to be integrated into patient-centric models. So for example, family and clinical history data that is very important are indispensable, and such integration will allow for a robust biological narrative when it is coupled to proteomics and metabolomics dataset. No doubt the challenges are many, including adherence to protocol standardization and data accuracy towards predictive outcomes. There is clinical genomic prediction suites and tools can be of use and further enriched to serve multi-omics data or serve as models to build new, more versatile ones. This paradigm will account for the interplay among the genome and the so-called environment in light of inter and intra-individual variability. So it will be genome, then proteome, and metabolome. So yet, for patient-centric models to be divided as much as possible of confounding factors and biases, sample size and population differences, all these needs to be addressed. For this, it's very important that collaborative platforms, argumentation, and data sharing among international networks and consortium become mandatory and necessary. So this is what now data integration BioAro is also working on that and developing our own platform. So I would like to thank BioAro staff, team, everybody on this, and I'm ready for the questions.
Thank you so much, Dr. Minal. We're grateful for sharing your knowledge with us. Now I can truly understand the importance of multidimensional clinomics and translational medicine. Dr. Minal will now take questions from the audience on the discussion we've had today. Please use your chat box for asking the questions or request to unmute if you would like to use your microphone. Well, Dr. Minal, as people probably just typing their questions, and we will give them some time. I actually have a question. So, given that we have so many different omics
Yeah.
that studying our genome, metabolomics, right? Uh, if, let's say, you are coming across a person and that person wants to know about all this scientific improvements, what would you suggest for that person? Let's say they want to know more about science in general, and they heard the word omics, and they're like, "Oh, what omics is?" If you can give a simple, clear summary of what omics is for that person.
Yeah, so if it depends on what kind of query and what kind of what they want to know, right? For example, simple, if something could be done using blood pressure measurement is can be done, or heart rate we have to check. We're not going to ask them to go for genomics and do the whole genome sequencing. So it depends what kind of query they have, and accordingly we can recommend them on omics. But yes, whatever the biomarkers or the assays which are available, for example, hemoglobin level and all this, just blood parameters. So we can go for the blood work and just check those parameters, and we can know about the health. But if we want to know the underlying causes, and then we want to see the heritable or inherited risk, then in that scenario we need to know the genetics, and then we will ask them to go for genomics. So again, even if it is the variants are present, the single nucleotide variants are available, could be possible that that person is not going to have that disease. There could be different epigenetic factors. There could be then we have to check for transcriptomic, for expression, gene expression. We have to check for protein level. So the first thing what we do is, obviously, we check for hormones, we check for metabolites, for protein level, and then to check the underlying causes, we go for genetics. So this is how it is.
We have a good feedback from people. Tell thank you. The presentation was really interesting. So we have a question that states how genomic tests can help physician in the treatment.
That's very good question, Sadhya ji. So yeah, um, depends. Again, it depends on what kind of thing we are looking at. For example, if we want to see in the family some disease is running, and if it is he- inheritable, and and we want to check that in fetus, definitely genomic test is going to help that physician to counsel them, to give the genetic counseling, and then talk about that, whether that fetus will be healthy or not. So in that scenario, you need to know the genetic variants. There could be some of the, for example, sickle cell anemia, or some of the disease in which you want to go for the genetic test of the person as well as for the sibling, so that they can help in bone marrow transplantation. So it depends. It could be help in diagnosis, it could help in treatment.
Thank you, Dr. Minal. Following this question, actually, I want to elaborate slightly further. So let's say there are some of the diseases that we treat only symptomatically. for example, rheumatoid arthritis, right? This
Mm-hmm.
is partially genetic disorder, right? That
Yeah.
can be prevented with the lifestyle.
Mm-hmm.
But let's say if the person comes and wants to know whether they will have rheumatoid arthritis in the future, can genetic testing help with that? And is there any way, let's say, we can prevent occurring of those disorders or diseases in the people?
Yeah, so that's good question. Actually, rheumatoid arthritis is not monogenic but polygenic disorder, right? So there are that's like many different genetic variants are involved, as well as different factors and immunocomponents are involved in this. Also, I would like to tell you that nowadays there is also transplantation for rheumatoid arthritis. So there is treatment where you can have your like T cells and all those things could be replaced, so that you have good immune cells, and then there is no rheumatoid arthritis in sort of treatment. So definitely, this we can help, we can find out uh, using genetic testing, that yes, yeah, or using in RA. Actually, we should, yeah, go for multidimensional approach, where you are uh, not only looking into the genetics but also epigenetic factor, or also protein and metabolites and everything. And now that nowadays there is transplantation is also available for the treatment.
Wow, that's actually amazing. I just briefly heard about transplantation. I didn't really know that it works with the rheumatoid arthritis. So it's a very good news to the people who are struggling with this
Yeah.
disease. And it's
Yeah.
unfortunately very common, especially in the elderly population. So
Yes.
knowing that we can cure it and
Yeah.
treat it, that's amazing.
Yeah.
We'll wait a couple more seconds for potentially more questions if anyone has, to type in a chat or request to unmute. And meanwhile we are waiting, I want to share with you, Dr. Minal, that we have a couple comments that telling that awesome presentation and thank you for the informative presentation. So we want to thank you very much for your co- hosting this presentation with us and sharing your knowledge.
Thanks to you, Elena. I just have one question, Alina. So in the beginning, was it not shared with everybody, the slides?
We could see only partially. It
Oh.
was, yeah. So luckily the issue was fixed. So, well, that brings us to the end of this webinar.
Mm-hmm.
Thank you for your participation and all your questions. I would also like to take the time and to thank you, our virtual audience who joined us from within Canada and around the world. This is incredible to see how diverse group of people joining together to explore science. Before we officially wrap up, I would like to take a moment to express our gratitude to Dr. Minal for taking time out and providing us with a really informative session. We appreciate your generously sharing your experience and knowledge on what is going on with our genetics, metabolomics, and our translational medicine. I encourage you all stay curious and keep learning and join us for the next week webinar with wonderful Dr. Preeti Balsundaram on how AI technologies come together with science. Please remember, the more we know, the better we're equipped to make informed decisions and improve our lives and the people around us. Thank you once again for being part of this webinar. Goodbye and see you next week.